Today's lecture is by Hasan DeMirci. Hasan got his bachelor's degree in Istanbul and then came to the United States to Brown University to work with Gerwald Jogl, one of the great experts on the structure of the ribosome. Well, whatever that is, but you'll find that out, I think, in the course of this lecture. So after he did his PhD work, he somehow got the idea that SLAC was the place that he had to be and packed up his world-beating collection of mutant ribosomes and brought them here. And tonight, he'll explain to you all what these things are for and what we can learn from them. So Hasan, thank you very much.
Can you hear me? All right. Thank you all for coming, and thank you, Mike, for the kind introduction. Today, I'm going to talk about mutant ribosome and the action of antibiotics. Could you raise your hands if you know what ribosome is? Who never took any antibiotic his entire life or her entire life? Never used antibiotic? Who? Nobody? Okay, so we know what antibiotic is, right? Anti means something anti. Bio means life, so it's anti-life, right? So before I start my public lecture, first I want to extend a public apology to my 4-year-old son. Right now I'm supposed to be with him before his nap time, but he's allowed to watch me until the first half an hour, so I really thank him for his understanding. So I'm going to talk about mutant ribosome and the action of antibiotics. In living cells, there's this tiny machine which slowly put the building blocks of life together and prints us in 3 dimensions. We have a genetic makeup, some sort of genetic information, and that information is somehow translated into this 3-dimensional life form, or a tree, or anything you can think of. Ribosomes pretty much prints the life in 3D. Today's lecture was supposed to be a tragic one, but with the surplus of my extra positivism, I can make it only maybe dramatic. So it's going to be a sad story. Here's the outline. First, I'm going to talk about what an infectious disease, a major problem that we're facing today. And I cannot talk about all the infectious diseases, so I'm going to pick one. And I'm going to talk about a major process of life, one of the most central concept of life, translation. How we translate genetic information to the protein world and makes us. So we're going to use the difference between our ribosomes and bacterial ribosomes, and we're going to preferentially kill the bacteria. Anti-bio means anti-bacteria. So we're going to hit the bacteria and not our ribosomes and kill them. To study the translation, we use a very powerful, very robust technique called X-ray crystallography. And then by using X-ray crystallography, we're going to try to understand how ribosomes work and how it functions. And we're going to have a little story about a drug called streptomycin and how streptomycin destroys the function of the ribosome. And I'm going to talk about future. But if you're at SLAC, future means today. So I'm going to talk about maybe a future elsewhere within the next 10 years, which is today at SLAC. So SLAC not only let us do very cutting-edge research, but also makes us feel like we're on the fast track. So we can do things that other people cannot do. Mike, that's why I am here, because this is the only place that I can do the research that I want to do. So in this photo, I show like a few people. If you look at the top row, you can see famous novelist. Emily Bronte just passed away after she wrote her first novel. She only was able to write 1 novel and nothing further. And here's a very famous physicist who got a Nobel Prize in 1933 for his contribution to the quantum physics. And these people died at relatively early age. And they died because of a disease called tuberculosis. And here is the drummer of the famous music band, Beatles, Ringo Starr. And some of you may think that he's not dead, right? Yes, he's alive. And he's partying. And one day I was driving to SLAC a couple months ago, and I was listening to my favorite morning show called Fresh Air at NPR. And maybe I will let him tell his story, how he's alive, what saved his life.
Oh, gosh.
So we're lucky to be here, Terry.
Yeah. Well, then you got sick again when you were 13.
I know. Well,
Tuberculosis, was it?
yeah, but that was from the area I lived in.
Industrial stuff?
Yeah. Where I lived, like, not every other home, but it was like, 6 or 7 cases in every street where people were just in the living room dying of TB because they didn't have a cure, of course. And again, God, shone his light on me in 1953 or 54 when they discovered Streptomycin, and that's what saved me. So they shipped me off to a greenhouse in the country.
A greenhouse? That's like a sanitarium?
Yeah, just a huge greenhouse. But instead of flowers, they put all his kids in there, and let us breathe some decent air for a change, and gave us Streptomycin. And a year later, I came out of there.
So how did you keep busy while you were sick? Had music entered your...
So fresh air and streptomycin. So I gave you guys 2 minutes of fresh air, and if you take a little bit of streptomycin, I think we're all going to be immune to tuberculosis. So before streptomycin, if you had TB, you're destined to die, right? And this drug saved millions of lives. And this is only one of the many antibiotics that we use. So imagine a life without antibiotics. I cannot. Imagine a life without Ringo Starr. What a disaster, right? So, and there are like a million other stories like Ringo Starr. And this thing is incredible. And this little molecules, antibiotics can save millions of lives. So I just didn't want to give statistics. If I tell you that 9 million people were sick with TB in just 2011, didn't mean too much if you don't know at least one story, right? Now we know there are at least 9 million stories. And among these people, 1 million of them are HIV positive. And just in 2011, 1.5 million people died because of TB, and half of them is almost HIV positive. Why I'm giving the HIV example? It could be HIV or cancer. When you get sick and you have immune depression or immunosuppressed immune system with some drugs, if there's no antibiotic while your immune system is depressed, again, you're destined to die. If you have an organ transplant, they suppress your immune system, and they give you some antibiotic. But during that time, if there's an infection, and that bug is resistant to all the antibiotics, then again, you have no chance, but again, you're gonna die. So these are the drugs, first-line drugs today used to treat tuberculosis. And if you look at it, the last one is introduced almost 40 years ago. And there's a 40-year lag, and this is the latest tuberculosis antibiotic. It took 40 years. So it's not an easy task to develop a new antibiotic. First of all, there's a $1 billion price tag. And for a pharmaceutical company, investing $1 billion on a drug that you cannot use regularly, it's not profitable, and they have shareholders. So what I'm trying to say is that it needs a public-private collaborative effort to tackle this problem. So government, academia, pharmaceutical companies, we should all get together and design new antibiotics, because if the last drug, vancomycin, doesn't work against these superbugs, we have no choice. We're going to go back to pre-antibiotic stage or period, and when there's an infectious disease, we're all going to die, right? So yesterday actually was World TB Day, and I'm here giving a public lecture, and I want to raise the public awareness about this disease. So we all know that we can prevent this disease in our life. We can design and develop new antibiotics, but we have to really collaborate all together, pharmaceutical companies, academia, government. Let's say we didn't do that. What's going to happen? We're going to go back to this old age. This is a cover of Le Petit Journal from 1912, and this is the disease called cholera. So at that time, an infectious disease means a death coming in, killing millions of people. So how are we going to fight with this bacterial antibiotic-resistant mutant superbugs? So we have to hit our enemy from one of its vulnerable points, right? It has to be a very central process that when you hit the antibiotic, the bacteria with the antibiotic, you should really hit something central so you kill him. So here's the central dogma of life. DNA makes mRNA, makes protein, makes us, right? So we have some genetic information. DNA is the book of life. And the information from DNA is copied to messenger RNA. There's a message. And that goes to the protein language. So what is ribosome? I want to really make sure that everybody knows what ribosome. Ribosome is this last step of this central dogma, arguably the most complex step of the whole life. It takes the message in the messenger RNA and translates into protein language. So in a sense, what ribosome does exactly what I'm doing right now. I'm thinking in Turkish and speaking in English. So in a sense, there are mistakes along this way, but the number of mistakes that you make over here is relatively very small compared to the ribosome. So I make a lot of mistakes. That's what ribosome does. It makes a lot of mistakes during this stage. So DNA is the book of life. If you want to make more books, you need a book printing machine, a giant copier. So then you have a small project, you use a Xerox machine, just a copy of a page, and that's your instructions. Now those instructions goes from DNA to the mRNA, messenger RNA, and these instructions are printed in 3D by the ribosome. Here it's a Yoda. I couldn't find a human, so apparently people like printing 3D Yoda. So that's what ribosome does. Use this information and prints three-dimensional proteins. So here's the structure of a DNA. That's where we store our genetic information. It's a very boring structure. It's almost mundane. It's a double-stranded helix. If you want to make more DNA, you open it like a zipper. And when you see an adenine, you put thymine. If you see a guanine, you put cytosine. Very simple. 4 letters. That's how we store our language or the genetic information. Unlike DNA, RNA has personality. So what I mean by that, it's just not stores the genetic information or take the message from DNA and takes to the ribosome, but there are different flavors of RNA. Some of them are structural. That's part of the ribosome. Actually, ribosome, which makes the protein, has 2/3 of RNA in its structure and 1/3 protein. So when they come together, they form the ribosome. And here in three-dimensional structure, it looks like more sophisticated, not just like a little stick. Again, similar language, but there's a difference. Instead of thymine, it's now a uracil. What is protein? Literally flesh and blood. These are like examples that I can find. These are the modern versions of the protein that you can buy over the counter. And here how it looks like in 3 dimensions. It looks like there are these rotini pasta. There's these arrows over here, and they're connected with this unstructured loops, right? That's what protein looks like. Different proteins has different orientation of these things. If the message is long, you can make a larger one. If it's short message, you can make a small protein. And again, you can see that the language is quite different. Now we're going to move from 4 letters nucleotide language of the mRNA world to the 20-letter language of the protein world. So how proteins are made? This is an 80-feet sculpture made by Mara Hazeltine. And this 80-feet sculpture is sitting in the garden of the Cold Spring Harbor Laboratory in New York. So this is an artistic depiction of protein synthesis. Proteins are manufacturing the proteins, right? It has 2 subunits, a large subunit and a small subunit. They come together and they start dancing on the messenger RNA. While they're dancing, they're making these beautiful proteins. So this is something we can see, right? Because it's big. It's a big sculpture. How we're going to see the tiny ribosomes in our cells, those tiny machines? We need a lens, right? So we can magnify it. But what if I tell you that there is no such lens? We cannot see ribosomes with a physical lens. It's so tiny. I'm going to come back to this later. But imagine, let's say we can see the ribosomes. I'm going to show this pseudo lens, or like an imaginary lens later, called X-ray crystallography. A protein or a ribosome. Now I'm going to show what a ribosome looks like if you can see it. This is magnificent wet noodle soup of what is called ribosome. That's where you make the proteins. In bacteria, in human, there are slight differences. We're going to take advantage of that differences later. So it has two subunits, a small subunit and a large subunit. So the small subunit is the brain of this machine. It deciphers the genetic information, and there are two arms that attaches the amino acids together on the large subunit. Those are the hands. So it's going to read and decipher and make the tiny proteins over here. So now we know how it works, right? Completely. So, okay, I'm going to split this thing open like a clam. So we're going to look at it from the interface, how this thing dance. So here's the crown view of the large subunit, and here is the small, this is chick view of the small subunit. Here's the beak, the head, the body, and the spur. Transfer RNAs, which is kind of base pairs with the messenger RNA and delivers the amino acids to the ribosome. They enter the ribosome from the acceptor site on both subunits. They move to the peptidyl transfer center, and they exit the ribosome from the E side. So they come, they move, and they exit. So in a sense, they're like the mailman. They bring the message or the envelope, they deliver it, and go. So now I'm going to show a movie. A beautiful choreography of this how two subunits dance. By the way, there is no such association or no such website. It's all made up. So this movie is made by Said Sanuga from MRC Laboratory, Medical Research Council in England. Here's the messenger RNA, and here's the brain of the ribosome, small subunit. And now this messenger RNA will base pair with the transfer RNA, which delivers the amino acids to the ribosome. And there are three watchdogs here. Make sure that the first binding is super accurate. If you don't bind the first code properly, then everything else is going to be frame shifted, and it's going to be gibberish instead of protein. Now we have the large subunit. The watchdogs live, and the first tRNA is on the ribosome with the message sticking over here. Again, the second tRNA is delivered by a factor called elongation factor something, and now it base paired here. It's the correct, brain says it's the correct one. There's a signal over here. The mailman is gone, and now the two tRNAs are linking the two amino acids together. Now we have to move this message a little bit so we can allow the next tRNA to come. There's another elongation factor something that moves that thing and use some energy while doing that. So now we're ready for the third tRNA. It's going to come. Ribosome is going to decipher the message. The messenger RNA is over here. And the anti-message over here, they base pair. This tRNA lives. And now there will be another. So now we're going to see the whole thing repeating a couple times. This is the message. tRNA comes, A site, peptidyl site, and exit site. And while they're coming, they're delivering the amino acids. They're linked by the hand, and they exit the ribosome. And this beautiful thing over here, which is growing, is the protein. That's how ribosome makes the proteins. So there is a stop. It cannot go all the way. There is somewhere in that message a stop. It says, okay, we came to the end, and there's a scissor, molecular scissor comes and clips that. Say, okay, now the protein leaves the ribosome. If you want to see something, there's a rhodopsin, a protein in your eye. That's how we're seeing this whole thing. And there's recycling factor. Now we're going to recycle the whole thing. We're going to split them apart, and we're going to start over. We're going to make more of these proteins or some other proteins. So ribosome has no personality. Whatever the message that you give him, it just translates and makes that protein. Everything is encoded in that messenger RNA. Is always this dance perfect? Always? Not always. So this is a classic movie, 1992, Scent of a Woman. Al Pacino here is a totally blind Army veteran. He cannot see. But he meets with his friend Charlie, a stranger in this high-end restaurant, Gabriela Anwar, and he asks her, would you like to learn tango, darling? And she says, right now? A blind man is asking you to tango with him. He says, I'm offering you my services free of charge. What do you say? She says, I think I'd be a little afraid. Of what? Afraid of making a mistake. He says, no mistakes in tango, not like life. It is simple. That's what makes the tango so great. If you make a mistake, get all tangled up, just tango on. Exactly. If you make a mistake, if ribosome makes a mistake, then it just goes on. So it only makes right amount of mistakes. If you don't make a mistake, then it's going to be a very robotic dance, very slow, very planned, right? But it's not going to look artistic. It's not going to look beautiful. So now we're going to study this molecular dance, and we need a model system to study this bacteria. Let's say an infectious bacteria. Who would like to work with something very dangerous? Nobody, right? Exactly. So we work with this bacteria, a close relative of this bacteria. This is Grand Prismatic Spring of Yellow Spring National Park. This beautiful colors painted by this bacteria called Thermus aquaticus. And we work with Thermus thermophilus. Why do we work with Thermus thermophilus? Because in year 2000, their crystal structure of their ribosomes is emerged. So you can crystallize their ribosome and determine high-resolution structures. So you can see everything in atomic detail. And our lab also switched from this regular workhorse of the biology, E. coli, to Thermus thermophilus. First of all, it's non-infectious. If you go below 72 degrees, it slows down. If you go below 55 degrees Celsius, it dies. So you can dump it to the nature, and it'll be completely. So when you go to shower and take a nice shower, you literally cover your skin with Thermus thermophilus. Okay, so one of the bottlenecks of biology is doing genetic engineering. If you want to engineer something, you have to put some DNA, modified DNA to the bacteria. And you cannot do it easily. But somehow, Thermus is like a vacuum. It just sucks the DNA that you give it to the Thermus. You just feed him with the DNA, and it takes it inside and puts it in its chromosomes. So that makes our life very easy. And then we know what we work with. Its genome is completely sequenced, so there's no surprises. And I work with ribosome, and this little bug is sensitive to almost all known antibiotics that are targeting to ribosome, with 2 exceptions. And half of the antibiotics are targeting ribosomes to kill the bacteria. So that's great. And we know the crystal structures of these thermophilic bacteria. How we're going to see this ribosome? I said there's no lens that we can use it to visualize these things, right? That lens is X-ray crystallography. We use this technique called crystallography to see things. What is X-rays? The X-rays are discovered by Röntgen, and he received the first physics Nobel Prize in 1933. And here's the first X-ray photo. I'm assuming it's his wife's left hand, and this is their wedding ring. What is X-ray? So X-ray is a light. So this is the light spectrum. We can see things between this visible spectrum. Anything with the wavelength between 400 nanometer to 700 nanometer, your eye can see, or anything bigger than 400 nanometer. But if you want to see things like atoms, proteins, ribosomes, viruses, you have to use, oops, I don't know what happened. I can take a sip of water. So if you want to see things like that tiny, you have to use part of the light spectrum which corresponds to that wavelength, so you can see them. We're going to see the atoms, and in that spectrum of light, only X-rays allows to do those kind of visualizations. And we do X-ray crystallography. So we need crystals. So first we have to generate the X-rays. So to generate X-rays, it doesn't matter. So what we do is we take electrons, an electron gun. Sounds very fancy. It's a light bulb, right? It generates electrons, and those electrons hit to a metal target. It could be a copper. And then once we generate those X-rays, they're very intense and very dangerous. So we make them very parallel and very intense, and we put a crystal, and X-rays shoot the crystals, and they diffract a little bit. But X-rays are very poor diffractors. So when you hit a biological molecule, they only diffract a little bit. So that's why we need a crystal. You can put like millions of the same tiny molecules in the same orientation and hit them with the X-rays, and they diffract to a screen, and you collect that diffraction pattern. Is it still not working? Okay. No problem. I'll continue. So how do we get the crystals, right? So first we grow bacteria, and then we use sonic waves. We just shoot them with the waves, and they just burst. And once they burst, we can use some computer-aided purification systems. We can choose whatever we want to crystallize. It could be a protein, it could be a ribosome. So we can purify whatever we want to crystallize, and then we put them in this plastic dishes like the one that you see over here, and we get crystals. And then those crystals look beautiful. But you cannot see it because it's right over here.
1 minute.
Okay. So those crystals are just remarkably beautiful, flawless. So for a parent, your kids look perfect, right? And for a crystallographer, our crystals, the ones that you cannot see, it's just remarkably beautiful. And then what we do is we shine them with the X-rays, right? But the X-rays are very damaging. So when you hit the crystals with the X-ray, they just burn. So what we do is we take those crystals and cool them to very cold temperatures so that we can collect data. And what we do is we shoot them with the X-rays at cold temperatures and then collect the diffraction data. What a diffraction data looks like is, now imagine it's a clear summer night. It's beautifully dark. You look at the sky, and what you see? Thousands and thousands of stars, right? That's what a diffraction pattern looks like. It's very complicated. So now what we're going to do is we're going to look at the sky and measure the distance between each star and look at the angles and calculate all of them. It's a very complicated computational challenge, right? That's what we're going to do. And we're going to use very powerful computers, and we're going to calculate the distances and angles between those stars. And then we're going to reconstruct the three-dimensional image of the biological molecule in that tiny crystal. So, again, you cannot see the visuals, but it's okay. Okay. All right, it's coming. By the way, you missed one joke while I was describing the crystals. Since I don't have the visuals, can I show it? Okay, excellent. All right, so where we left? Over here, we use X-rays. As I described, we put the crystal, shoot them with the X-rays, and collect the data on a detector. So how do we make the crystals? We grow bacteria. We burst them open with the sonic waves, high-frequency sound waves. Once they break open, we can purify them by using our computer-assisted purification systems. And these are the crystallography trays, how we obtain the crystals. And I brought an example of plate here. If you want to see how a crystal looks like, I can show them on the microscope right here. So then we get the crystals. They come in all different flavors. They look beautiful. They have like this cubic shape and all sort of different shapes, trigonal, pyramidal, and some hexagonal ones. As I said before, for a family, like you think your kids are perfect. For a crystallographer, our crystals are perfect. And this is my son. He looks perfect. Okay, then we cool our crystals at liquid nitrogen to cryogenic temperatures and shoot them with the X-rays, these tiny machines, if you have a university lab, that's what you usually own. And then the diffraction pattern, those tiny spots, the stars that I described. But we're lucky that we're at SLAC, so we don't need these tiny machines. We have the entire synchrotron and world best laser, right? Why do we have to do this? So now again, I want to compare one more time how we see with the human eye and how we see with the X-ray crystallography. So this is a plaster model, a palm-sized plaster model of the ribosome. So the light, the visible light, reflected from the object, and the human eye can focus that reflected light to the retina, and there's a protein called rhodopsin that's made by the ribosome that lets you see. And when you do X-ray crystallography, here's the real ribosome in the cell, a tiny molecule, but we crystallize it to amplify the signal. I described that the biological molecules are poor diffractors of the X-rays. When you make them multiple copies, the signal gets amplified so you can detect it better. So now, from 3-dimension, we're on a flat surface. X-rays are waves, and when the waves hit the screen, you only see the height. We cannot see the wavelength, right? We don't know how big is the wave, only the height. So then we have some prior knowledge about the object that we study. If it's a ribosome, we know the sequence of the RNA and the protein. If it's only protein, we know the sequence of the protein. So now, this is our lens. This box here, the Pandora's box, this is our toolbox. This is X-ray crystallography. And we do somehow some magics and use all of those angles and distances and reconstruct this 3-dimensional image. Who wants to see what's in our Pandora's box? I'll give you a split-second view of what's in the box. We're going to see under the hood, but for a very short period. This is what's in the box. Okay, So this is the passion of my life. After all of these, we calculate this electron density maps, this beautiful electron clouds. How beautiful is that? That's why I started doing X-ray crystallography. When I was a college student, the 3rd chapter of my biochemistry book was a beautiful electron density map of a protein. I said, I want to do this when I grow up. Okay, so I said we have some prior knowledge, right? We know the sequence of the object that we study. You see some continuous parts over here and stuff sticking out. So first, we built the main chain of the protein. So we put these backbones of the protein. Remember, ribosome makes the protein that literally connects these blue bones. So they glued on the ribosome together, and they extended like this. Oh, by the way, there are some extra pieces over here and there, right? Those are the side chains of amino acids that gives the amino acids their personality. So this is a tyrosine, this is a leucine, and after you finish building the model, sometimes you can see some water molecules. It's an oxygen. So how powerful is that? This distance over here is only 1.5 angstrom, 10 to the minus 10 centimeter. So it's like very, very tiny distance. By using this X-ray crystallography, we can see these beautiful things, right? Now you know what is X-ray crystallography. And you saw my model system. Now we're going to study mutant ribosome, those scary things. So the main question that I want to study is, what do antibiotics and mutations in ribosome tell us about the decoding? What is decoding? Deciphering the genetic information on the ribosome. I cannot talk about all the antibiotics, and I cannot talk about all the mutations. But first, I want to talk about what is decoding, right? So we saw that beautiful dance of the ribosome, but now we're going to use this powerful technique and look at everything in atomic detail. We're going to see every atoms, every details of this book of life, and we're going to see how we read the book of life. Very fundamental process. To me, it's the most fundamental biological process, reading genetic information. There's nothing more fundamental than this. So here is the bacterial genetic code. And you can see that each triplet over here, encoded in the messenger RNA, is code for an amino acid. And there are certain peculiarities about this genetic code. Let's focus on this box over here. This is called a degenerate codon box. Ribosome somehow monitors the first and second position, so it's almost non-changeable. You cannot change. But the third position doesn't matter really what it is, right? Either 4 of the letters still codes for the same amino acid. How does ribosome know where's the first position, where's the second position, and where's the third position? So the translation is very fast. It's almost 20 amino acids linked together on the ribosome with the accuracy of a mistake 1 in 1,000. So after you make 1,000 peptide bond, there's one mistake. That creates the drama in our life. Otherwise, if you make no mistakes while you're making your DNA or messenger RNA, it's going to be a Groundhog's Day. Every day is going to be the same. No changes, no evolution. Mistakes are good, just the right amount, okay? So this is the messenger RNA, and this is the code. This code is specific for an amino acid called phenylalanine. And ribosome somehow brings the thing to close proximity. Biology is all about proximity. You put things together, right next to each other. And that's what ribosome does. Pairs the messenger RNA, which is transcribed from DNA, and this adapter molecule called transfer RNA base pairs with the messenger RNA on the ribosome, and there is an amino acid attached to the 3' end. You don't even have to know the number. So ribosome, if A base pairs with the U, this is the correct base pairing. A always base pairs with U, and G base pairs with the C. But you see that there's this G. It's a mistake on the third position, a GU mismatch. But ribosome thinks this is correct because it's in the first position, or third position, sorry. If you make the same mistake, GU, in the first or second position, ribosome knows that these tRNAs are wrong, and they reject it. They're not part of the protein that you're making. So how does ribosome discriminate between correct and incorrect tRNA? This is a very simple slide. This is how ribosome does it in a 2-step kinetic proofreading model. How simple it is. I'm going to make it even more simple. So now you're the correct tRNA, right? You're over here. And this is a 100-meter Olympic hurdle race. And these are the hurdles that you have to overcome to make this peptide bond over here. There are a bunch of tRNAs over here. They're going to start racing. But the race is not fair. For the correct tRNA, the hurdles are so short, maybe a few inches tall. And the correct tRNA somehow goes to the other side by dancing and forms a peptide bond. But if you're the wrong tRNA, the number 7 is the correct tRNA. But if you're the wrong tRNA, the hurdles are like Great China Wall. So you somehow have to jump over like Great China Wall 6 times to go to the other side. And the correct tRNA comes fast and wins the game. And it's a great athlete. He can run this race 20 times per second, and he wins the game, the correct tRNA, or maybe loses the game once in 1,000 race. How great is that? So now, this was the simplest version. Let's look at the atomic details of this thing. So first, the tRNA comes and binds to the messenger RNA on the ribosome. So now I'm showing the center of universe. This is the brain of the ribosome. I'm deleting everything, and we're only seeing the messenger RNA. And this is the code, 1, 2, 3. Just 3 codon. Ribosome brings the tRNA and put them right next to each other on the ribosome's brain, the decoding center. So ribosome now is going to look the position 1, 2, and 3. And it's going to use a very elegant system, an elegant mechanism. So these great things are part of the ribosome. When you have the correct tRNA and messenger RNA together on the ribosome, they flip. Now imagine 2 fingers. And now these 2 fingers going to monitor the isosteric nature of this codon-anticodon base pairing. What is isosteric? Iso means same. Steric means geometry. So there is a geometry here. Now I have to describe this. A and G is my point fingers, and U and C is the pinky. So A has to base pair with U, and G has to base pair with C. When you do that, the distance is always same. If A base pairs with the G, or C base pairs with U, it's either too long or too short. So we're going to measure that distance. It doesn't matter what you're measuring, right? If the distance is same, ribosome is going to say, this is correct. So it's not even looking at the bases. It's only measuring the distance. What a simple mechanism. But it only does it for the first and second position. Those fingers only fit if there's a correct geometry in the first and second position. But the third side, the third position is monitored by an angle. So it doesn't really matter what's over here. Actually, it does, but I'm oversimplifying it. It lets the base pairing in the third position more easily. So it brings us to so this is a beautifully choreographed dance, right? We only see the first step. After this codon and anticodon is the correct one, ribosome has another move, a large, big move. So that brings us to the activation stage. So this was the center of life. I'm going to put it back where the ribosome is. Now we're looking at the brain right over here. And this was the chick view of the ribosome. We're going to cut the head of the chick, and we're going to look at it from the top. So ribosome goes from an open to a closed transition if there's a correct tRNA. Maybe I should play it. So this is the correct messenger RNA and correct tRNA. So ribosome does this during the decoding. So this is a very optimized motion. I said ribosome only makes one mistake in 1,000 peptide bond formation, right? This is like a kind of adjusted that 1,000 per one mistake. But you can make mutations in the ribosome either over here or over here. If you make mutations here, that favors the closed state. Now you can close even for the wrong tRNAs. And now you start making more mistakes. And this mutation right over here favors the open state. Now you even reject the correct tRNA. Now ribosomes are hyper accurate. Makes only one mistake in 1 million. But would you like to stay here for another 10 hours for me to speak slowly and make less mistake? Or you just want to get out of here in 1 hour? That's the question, speed or accuracy, right? Now ribosome is slow, but it's more accurate, but we don't like that. There's a fitness cost. It costs this bacteria being very accurate, slows it down. So now I put it back. Before it was open, close, we were looking from top. Now that motion is like this, and tRNA binds here, and it clinches on the tRNA, closes over the tRNA. And the motion is from orange to green. So now I'm putting the full-length tRNA so we can see the whole thing. There's another tRNA at the peptidyl transfer center. This is where the arms come together and amino acids are linked, but they're far apart from each other, right? We have to bring them together, and this is the exit site. And tRNAs are delivered by a mailman called elongation factor. And this domain closure activates this factor, and it leaves. That's the step over here. And when the domain close, that kind of reaches to that mailman and say, okay, you delivered the right tRNA and you can leave. And that's what happens. Now I'm putting the large subunit and putting the whole picture together so we can see. So now the mailman is gone, and the tRNA has two options. If it's the correct tRNA, it's kind of clinched by the ribosome, stabilized on the ribosome. If it's the wrong tRNA, there is no domain closure, and the tRNA flies away. That's called proofreading. But if you're the right tRNA, you're stabilized by the ribosome, the mailman is gone, and there's a remarkable torsional strain. It's like a spring. Swings into the peptidyl transfer center. And Shazam. That's how you make the peptide bond. Now you put the two amino acids together. Okay, the brain deciphers, and you make the peptide bond. Now we know what is decoding, right? Let's go back to the question again. How antibiotics, or what antibiotics and mutations in ribosome tell us about the decoding? So now I'm going to talk about de-antibiotic and de-mutation. Just two examples, because it's going to be like another 10-hour lecture if I talk about more. So this is just a simple sentence from this book called The Molecular Basis of Antibiotic Action. This is the Bible of antibiotics field. It's 40-year-old and written by really famous people. And in this book, they claim that the diversity of effects produced by this aminoglycoside and the ingenuity with which they have been pursued have made the streptomycin story the most fascinating and complicated one in the field of anti-ribosomal action. So I'm going to tell you a fascinating story. I maybe contributed very little, but I'm proud of that. So this streptomycin, this drug, is really amazing because when you give this drug to the bacteria, you kill the bacteria. If bacteria somehow, the DNA, while they were replicating, made a mistake and gain a mutation, become resistant to this drug called streptomycin. But there's an extreme case of resistance. Bacteria doesn't know that. Bacteria thinks that now I'm resistant. And you take the drug out, and the bacteria dies. Without knowing, it became dependent on the drug. It became an addict. And I'm going to talk about those junky ribosomes. Those ribosomes are the mutant ones. Okay, so this is a classic paper by Julian Davies et al. And this paper shows that the ribosome has a decoding center that deciphers the genetic information and monitors the codon-anticodon base pairing. And the drug streptomycin increases the error rates of the ribosome. So this paper shows that this antibiotic is targeting the ribosome. Okay, here's the brain of the ribosome, and here is the drug called streptomycin. You can see that if this is the brain, and these are the two fingers that I was showing that was monitoring the first and second position of the code. And now you're putting a drug right near your brain, and that kind of makes you fuzzy. Or now drug pulls the brain of the ribosome and squishes the brain. And now your brain doesn't work anymore. With the drug, when you put the tRNA, remember when they swing, they have to swing to a perfect spot to look at the first and second position. Now you distorted the brain, it's swinging but has no clue where it's swinging. So there are two types of antibiotics, bactericidal and bacteriostatic. And streptomycin is a cidal one. So the bacteria commit suicide when you give them streptomycin. So they keep translating the messenger RNA and makes gibberish protein, and they hoard all of those nonsense stuff they produce. And after a while, they notice that full house full of junk, and they die. And static ones kind of, you're putting a boot to your car, and you cannot drive the car. But when you take the boot, the parking boot, the car can go again. So those drugs that I'm not going to talk about. And static ones are not the interesting ones, but this is a cidal drug. So that kills the bacteria. So that's a good one. So now I'm going to show another movie. This time I made the movie, and it shows like what streptomycin does to the ribosome. So the streptomycin binds right over here, and it distorts the brain. And also there's a support structure. So the brain has two fingers, right? And there's a support structure that holds that two fingers in position. So it can flip very frequently without changing its direction. So this support is destroyed when you have the drug streptomycin. Not just distorts the brain, but also increases the plasticity of the decoding center. So there is this nice support and network of chemical interactions, and those are destroyed by the drug. And now, again, the brain becomes very flexible, and the two fingers just swings in all different directions. So if I summarize what I said so far, streptomycin distorts the decoding center of the ribosome. And now I'm going to talk about D-mutation, the one that makes the ribosome drug junkies or antibiotic junkies. So I'm a crystallographer, and this is what a regular ribosome crystal looks like. So it looks like a rod, long, and kind of, tetragonal. But once you have the mutation, now they look very different. Okay, so now we're in a stadium, and we have 3,000 people. And we're supporting San Francisco Giants, okay? Only one person comes and replaces one of the audience, and now we're supporting Boston Red Sox, okay? This is Giants, and this is the Red Sox. how that one person can affect the whole audience, right? How much personality is changed. So this is one simple mutation. But if I take the drug and give the drug to these mutant ribosome, it crystallizes back like a wild type again, the normal ribosome. So the drug stores the functional state. And then we replace another spectator with a second person. It's called a suppressor mutation, a second mutation on the ribosome. And now we're supporting SF giants again. And that's called the suppression of the mutation. And now you're released from that dependent state and become independent SF giants fan. So what's the difference at molecular detail? So by just looking at the crystal, you can see that which one works and which one doesn't work, right? So I'm a crystallographer, and in crystals, you have multiple copies of the same molecule. So they're like tiles. You just put them together. In the normal ribosome crystal, these two tiny molecules interact with each other, and in the mutant one, these two ribosomes slide over this edge, and now this protein touches to the beak right over here. In crystallography speaking, this is like 2-mile difference. So it moves like quite a bit. Now I'm showing this mundane, boring, Val-type functional structure at atomic resolution. And this is where the mutation is. That one mutation is going to make ribosome dependent on the drug. And we calculate the experimental electron densities, and these clouds over here is the new electrons and the positions of the new structure. And this tiny thing over here is the mutation. That scary poster, that purple mutant, is this thing. So you can see that the new map tells us that the brain is distorted. Let's put the cartoon again and move the electron density. So the drug was pulling this brain over this direction, and the mutation distorts the structure the other way. So if you move the brain over here or pull it back this way, it doesn't work. But when you put the drug, it pulls it towards the normal site, and now you function again. So either the drug by itself or the mutation itself kills the bacteria. But when you put them together, it's fine. I know this ribosomes function, but not like the normal ribosome. So there's a fitness cost comes with this mutation. So imagine a dog running with his forelegs versus just walking in his hind legs. So this dependent ribosome function, they walk, but not as good as the Val-type one, the regular ones. So if I summarize, the drug-dependent mutations result in extreme distortion of the decoding center, and the physical presence of the drug partially restores the deformation of the decoding center. So it makes it functional, but not as good as the regular wild-type ribosomes. So now I'm going to talk about today. The future was today here at SLAC. So this is our 3-mile tunnel, the new free electron laser. That's where we do our experiments. And we're right in this building, right over here. And this is the synchrotron, the Spears at Stanford, where we do regular crystallography. And electrons here injected right over here, they travel almost a mile, and they enter this part of the tunnel called undulator hole. And these are 100-meter nice magnets. They align with super accuracy, maybe less than a millimeter on height. So it's almost an engineering marvel, maybe thought to be impossible to build. But now electron travels this 100-meter of alternating magnets, and when they exit, they generate the Xrays, which is 1 billion times brighter than any of the third-generation synchrotrons can generate. So for an X-ray crystallographer at SLAC, the future is 1 billion times brighter. So, and then we take those X-rays and use them here at the final station called Coherent X-ray Imaging End Station. That's where we shoot our crystals. So now we have very bright X-rays, and they're super damaging, right? And now we can package those bright X-rays into very short pulses. These are 50 femtosecond pulses. A light can travel from here to moon in 1 second. all the way to the moon. And in 50 femtoseconds, light can only travel only thickness of a hair. What is 50 femtoseconds? Femto means 10 to the minus 15. 1 billionth of a billionth of a second. So very short time, you can package the photons and shoot them through the sample, either a molecule or a crystal. So this technique called diffraction before destruction. So I have my PhD and postdoc from Brown, from a small state called Rhode Island. Okay, so now you're driving a very fast Italian-made car with 300 miles per hour, and there's a highway patrol at the entrance of the Rhode Island. They're waiting for overspeeding people, but when you enter with 300 miles per hour, by the time when he notices that you're in Rhode Island, overspeeding, you're in Connecticut. It's too late. And that's exactly what happens here. The X-ray comes, gets in, and exits. But before the damage starts, you already collected your data. You went in and out. And then it explodes, but it's already late. You already collected your data. So I came here to work with a former staff scientist, Mike Bogan. And in his lab, we did our first not time result, but this is called like the serial femtosecond diffraction. So now we don't have to freeze our samples because we can outrun the radiation damage. That's why we were cooling stuff, to make them overcome that radiation damage, the heat. So since everything is in liquid phase, we can do things in like normal life. In cells, there's water. Now we can do things in water. So this was the first data that we collected in March. And now, here's the future. Now we're planning to not just use a single capillary to put the crystals to the X-ray beam, but we can use 2 capillaries within each other and send the tiny crystals and mix them with the antibiotics, decoding complexes. Like, I showed a model of decoding, but that's still a highly disputed topic. I showed my favorite model, and there are still discussions about like which model is correct. So now we can study those kind of things in time scales that we couldn't penetrate by using frozen crystals at synchrotron. So we can do millisecond mixing, and I said like there are 20 peptide bonds in 1 second. That means like we have 50 milliseconds to look at 1 peptide bond. So I cannot freeze a crystal in that speed. It's already second. It's done. It's over. But with this technique, we can do things that at very short time scales. And that's called kinetocrystallography. So we're opening up a whole new field. So far, I showed a lot of movies, right? We had great fun. But those are not scientific movies. I showed science and fiction. So for example, we take a snapshot of a photo of a ribosome, and we put the drug, and we take another photo. And we think those movies, for example, in the first one, Sean White looks like this. In the second one, he looks like this. So what we did is we interpolate. We just imagine what could be the motion, right? It's very simple. That's what we did so far. That's where we are today with the current technology. But is it like that simple? Is that biology that simple? I want to finish with a crescendo. That's what I'm talking about. That's what biology is all about. We're going to see all of these cool stuff, the cool biology, which we couldn't penetrate with the normal crystallography by using this brilliant microscope that we have at SLAC. And these are the army of people who helped me to get to this point. Currently, I work with Mike Soltis from SSRL and Jody Puglisi from Stanford, and my PhD advisor, Gerwald, and Al is my postdoc advisor, and Steve, he's a great scientist who helped us to get all of those cool mutants, even the one that I showed you today. And Mike Bogan was the person who brought me here, and now he's left. And these are great people right now in our lab, these colleagues, Ray, Hartawan, Claudio, DeMirci. And I would like to also give a very special thank to Venki for giving me the wonderful opportunity to visit his lab at MRC. And it was Ann Kelly who showed me how to crystallize the ribosomes. And Frank helped me to collect the data, the ones that I showed you. My current research is funded by Stanford Institute for Chemical Biology seed grant. And before I was funded by NIH and NIGMS. So the research that I showed you is where the taxpayers' money went. And I hope the public is happy where the monies are spent. And with that, I'd like to thank you all of you for listening to me. And I'll be very happy to entertain if you have any questions.